Okay, so the paper that we're going to talk about today is from last year. I don't mid-year sometime, maybe we can look it up. Cell paper from the Rasmussen group. These guys have been doing ancient DNA for a long time. So, for example, one of the things that they've worked on for a long time was cracking open teeth from mass graves in Europe. Graves that were put in place during plague epidemics. There have been 3 big epidemics of Yersinia pestis in Europe. The modern one, which until about 100 years ago was a big deal in Europe. The Justinian plague, and the one before that, whose name I can't remember. Third pandemic, plague of Athens. These plagues devastated Europe. There's a story from London describing this guy walking around through the city, and there's nobody there. It was literally abandoned. Imagine a plague that, over the course of 50 years, reduces the population by 30%. Pretty awful stuff. There are parts of Norway, parts of Iceland, for example, and so forth, that were just completely destroyed. The human population was. They really were bad epidemics of the sort that we have no comprehension of anymore. Caused by Yersinia pestis, the plague. Yersinia pestis is, of course, fed by fleas, and so rats carry the fleas. The rats move around, the flea bites the rat, the flea picks up Yersinia pestis, and when it bites you, the fleas can't live on your body permanently, but they're desperate, right? They're little insects. And they bite you, and they can't productively feed from you, but they've inoculated anyway. And so you get the plague, and it's good. Plague comes basically in 3 versions. There's bubonic plague, where the organism is growing in lymph nodes in your body. There's a systemic infection, and there's pneumonic plague, where you breathe in the organisms and it grows in your lungs. It turns out, so the Rasmussen group has been working on this for a long time. They developed these really cool techniques of pulling teeth out of skeletons. Typically, these are unerupted teeth, so they're children's teeth. That means they're not exposed to the environment. Scrupulously clean the outsides of them, embed them in plastic, split them open, and then take the pulp out of the middle, and they're looking for Yersinia pestis DNA inside. This works because Yersinia causes a systemic infection, right? The cells are floating around in your circulation, and so when the person dies, some of those cells happen to be in the veins and arteries in your teeth. And they're trapped there. And teeth are like little time capsules in dead bodies. Teeth last longer than any other part of an animal's body during decay. And the root of the tooth has a little, just little holes for each root that kind of seal off, and they don't really just rot away like other tissues do. They tend to there is some decay in them. Some organisms get in there and grow, but they're fairly pristine inside. And so they're a great source for this kind of thing. These folks previously had shown that these 3 big plagues were all caused by Yersinia pestis. They identified the strain of Yersinia pestis by sequencing genomic DNA from these organisms, or from the DNA extracted. And with other mechanisms, other sorts of things, they've been able to follow genetic variation in Yersinia during the path of these 3 plagues. But all of these are caused by a particular strain of Yersinia pestis, strain orientalis. And those go back about 800 years. That's the time frame we're looking at. This paper goes much deeper in history than this. I'll point out too that the major concern about their previous work was about contamination. Those of you who've done PCR experiments know that if you set up a PCR reaction and you put primers in, the primers are going to find something. And particularly the way these guys did it, where they, in some cases, would do 40 rounds of amplification and then take the product, they couldn't see anything in it, and they would amplify another 40 rounds from that. And the danger of contamination is awful. They took extreme measures to prevent this. I mean, total disinfection of the outside of the tooth. They would do it in a clean room and make sure to do everything they can to make sure that what they collect is only from the inside of the tooth. They actually went through the process of setting up PCR reactions where you only use one set of primers once, and you never can use them again. Because if that DNA is around, it will find it. They were doing experiments. They were having colleagues doing experiments in other countries because of the danger of Yersinia pestis that they had in the lab. There's DNA, right? And so the primers would find stuff. So they took extraordinary lengths to prevent that. In this paper, they use a completely different approach. They don't do any experiments at all. They piggyback on some experiments that other folks were doing for a different purpose. And it's really cool. So let's see. So they piggybacked on a group of folks who were looking at population genetics in humans from ancient samples. And so they were interested in the population genetics in Europe. And they're looking at old museum samples, going back and opening up the teeth, taking out the remains of the pulp and doing human genome sequencing, right? They just sequence everything they can from the inside of that tooth. And what they were looking at, of course, was the human DNA, and they were looking then at how the human populations evolved in Europe over time. Interestingly, they find that when I think of places like, well, anywhere really, what you normally think of is that people spread out to these places, and then those populations more or less are the same forever, right? That is, if you go to Hamburg and the people there, you can follow their family history, and the family's always been in that part of northern Germany. That turns out to be completely and utterly false. Populations, at least in the area they were looking at here, it's absolutely clear that there have been mass migrations back and forth all over Europe many, many, many times. That people have moved around a lot in Europe. The paper is framed in the sense that they, having seen this, that they were asking the question, okay, why were people moving around so much? Maybe it has to do with plagues and epidemics. And then decided that given the Black Death is so important in Europe, let's look to see if we can find evidence of that. I suspect that the opposite is true. That's one of those stories you make up when you write a paper to fit everything together. My guess is that they had all this DNA, and one of the grad students working on the human project said, hey, we learned how to extract these teeth from the folks doing Yersinia. I wonder if there's any Yersinia DNA in that. And did a search through their data and found it by accident. Certainly they found other bacterial chunks of DNA, but interestingly, out of 101 Bronze and early Iron Age individuals, they found 7 teeth that had Yersinia DNA in them. That's pretty striking to me. I mean, really, 7% of the people were infected with Yersinia during these times? That's well, I guess you could argue that 7% of the dead people had Yersinia. Maybe that's a difference. And they span almost 2,000 years' time. It's pretty remarkable that you can get meaningful DNA out of somebody buried in the ground, from 5,000 years ago, right? So what kind of Yersinia DNA was this? Interestingly, they did see differences in essentially you can get nearly complete genomes of Yersinia from this these samples. And that means you could look at those Yersinia and see how they might have worked. And what you see is that the strains that, and all of these are different than modern strains, including Orientalis that caused the older plague, the historical plagues. And they're different from each other. And clearly there's an evolution then of this organism through these plagues. I would argue, I don't think they make this case, but I would argue that this isn't really Yersinia pestis that far back in time. Because if you look at the phenotypes they're looking at, this organism, if you found it today, you would call it a new species of Yersinia. It's most closely related to pestis. It's quite a bit away from things like Paratuberculosis and other strains of Yersinia. So it's clearly a close relative of Yersinia pestis, if not a direct ancestor, probably not a direct ancestor, but maybe close. And there's some really interesting aspects of the genomes, particularly of the earliest skeletons. Number one, they lack the gene required to grow in insects. So they would not have been transmitted to humans by fleas. That implies a completely different life cycle and a completely different mechanism for causing plagues. So this is the Yersinia murine toxin. I don't know why they call it that, but so it raises the question: how could they have been? How would you transmit it? I mean, today you get it from fleas, but or you inhale it, and so it could be that inhalation was the primary. Of course, yeah. Well, the mice carry it, but it's the fleas that bite them, and they're the ones that transmit it to the humans. Yeah. Like hantavirus or something like that? Yeah, sure. And that's unclear. The other thing is they lack the flagellin gene, the flagellar genes. Excuse me, they have the flagellar genes, which are absent in the modern strains. They're absent because that's the big serological handle that organisms have to fight it off, right? That's a giant target. That's a big billboard that says, I'm an invader, macrophage, come here. And so modern strains lack flagella. These ones have it. And so this implies that it could not live in lymph nodes. That implies that this would not have caused bubonic plague. And so you're looking at pneumonic plague or other kinds of infections for this organism. Very, very different kinds of diseases, right? I think this is interesting because a lot of times when people go back to historical records and try to identify what a particular plague might have been, you always rely on kind of the symptoms and the epidemiology in the local environment. And a plague caused by this organism would in no way, would never have been considered a case of bubonic plague or Black Death. Because the symptoms wouldn't be the same, the mode of transmission wouldn't have been the same, and people wouldn't know what caused it. Those historical records, by the way, are terrible. It's really, really hard to figure out what an ancient epidemic is. They didn't use the kind of terms we use today, a lot of superstitions. It really wasn't bad air from the salt marsh that causes malaria, for example. And there are differences, particularly in the flagellin gene, throughout in the skeletons based on time, at least, where the genes are becoming less robust. So what they say they want to do, so what they've got then is kind of a historical record of how Yersinia pestis and the Black Death emerged. And it emerged recently. Recently in the sense that the plague, the way we see it today, is something from the last 1,000 years, as opposed to the antecedent diseases have been around for a long time. They'd like to look at other geographic regions. The modern strains, and by modern I mean the three known pandemics of plague, originated in Asia and spread to Europe and Northern Africa and North regions from there. So Asia would be a really interesting place to look at the different strains and how they emerge. Turns out rats are not the primary host. There's some kind of a marmot or something that it lives in naturally in Asia. I'd like to see more teeth. 100 teeth and 7 positive for Yersinia is not a lot to go on. I'd like to see some where you can tag the humans to particular plagues, right? Plague of Athens. I'd like to look through this for more than Yersinia. What about viruses? I mean, essentially any infection that goes through the circulation, you want to be able to detect this way if it's got a genome in it. That means not tuberculosis, but maybe not syphilis. There is a nerve in there, but I don't think those nerves normally get syphilis in them. But there's a bunch of other diseases you can test this way. What about typhoid fever? Typhus. What about, I don't know, herpes, something, I don't know, what, you can imagine anything, right? I'd like to sift through all the bacterial and viral sequences they pull out and see what is in that. I think that'd be really cool. Yeah?
Why?
Because that's usually all that's left. Of course, the soft tissue decomposes rapidly. Most of the bones don't last that long either. The skeletons they get infiltrated with microbial growth, and all the organic material gets degraded and so forth. Teeth, though, because of the enamel and just the hardness of the bone there, are like little time capsules. And for a lot of populations, all we know about them is from the teeth. That's kind of a standard archaeological. The first thing you find about new people are teeth. So, what I'd like you guys to do is to have a look at this information. The paper is here. You can click on it and have a look. There's a little accessory thing that shows you. We've got some. So here's what a skeleton looks like. The bones are pretty awful, pretty awful, but the teeth are good. What else? They've got the main chromosome from these 7 teeth. They've got the plasmids that are in the cells from those teeth. And there's a bunch of other data in there. There's some phylogenetic stuff in there. Have a look. And I want you to come up with something that strikes you. And again, I think a good prompt for this kind of thing is, what problems do you see with this? But if you were a grad student in this lab, what would you tackle? What would be something that you found interesting enough that you would say to your PI, Dr. Rasmussen, I'd like to do this. I'd like to find this out. Does that make sense? Good job.